Jak Stat are the Janus kinase signal transducers and activators of transcription pathway is a receptor signaling cascade pathway. Jak Stat is a principal signaling mechanism for cytokines and growth factor. Jak Stat pathway will result in the gene activity in a cell. This gene expression can stimulate cell differentiation, proliferation, migration, and induce apoptosis. It can regulate various cellular stress, including hypoxia, reperfusion, endotoxin, ultraviolet light, and hyperosmolarity. A variety of ligands can activate the Jak Stat pathway. The ligand needs to bind to the Jak Stat specific receptor expressed on the cells. Jak Stat receptor consists of a ligand binding domain and a cytoplasmic tyrosine kinase domain. When ligands, for example interleukin-6, bind to the binding site, the receptor will dimerize, and Jak tyrosine kinase domain will phosphorylate itself. This phosphorylated receptor attracts the STAT molecule. The STAT molecule will be phosphorylated by Jak at the tyrosine residue near the C terminus. The phosphotyrosine will allow two STAT molecules to dimerize. The activated dimerized version of STAT can enter the nucleus via a nuclear import pathway. The dimerized STAT will find the response element gene sequence, the regulatory sequence, to activate or suppress the specific gene expression. mRNA will be transcribed and translated into proteins. It is important to regulate the Jak Stat pathway as overexpression of regs will lead to abnormal metabolism of cells. There are three types of negative feedback mechanism to regulate Jak Stat pathway. One of them is the suppressor of cytokine signaling proteins or SOX proteins. SOX proteins are recruited to cytokines to initiate a negative feedback loop to switch off the Jak Stat pathway. SOX proteins are constitutively present in cells at low levels. They recruit to the same Jak receptor as the ones that activate cell metabolism. However, when ligands such as interleukin-6 bind to the receptor, the whole process of the Jak Stat pathway will initiate, but in this case, the regs are the SOX gene. The SOX gene will be expressed, and SOX proteins are produced and then interact directly with the Jak. They can inhibit the signaling pathway to which it recruited them initially. SOX2 will bind to the receptor phosphotyrosines and inhibit signaling by competing with STAT molecules for recruitment to the receptor complex. STAT molecules cannot be dimerized without this phosphate group, and monostat cannot enter the nucleus as transcription factor, hence no expression of the response element.